I presented my last paper about betaine in July, and that was the paper that give me the possibility to be here today. So I want to thank Amber to contact me after publishing this paper, and also to push our research not only on the GAT1 wild type, but also to look what happened with the mutants. So we start to investigate the mutant behaviors only 6 months ago, but today I want to present some of these results and making some and point out what we find on GAT1 wild type and betaine. Oops. It's not okay. Oops. Okay, first of all, I want to underline that betaine is a trimethylglycine, is a natural compound, is stable, and importantly, is a molecule that can be found in any kind of living organism. So, and for this reason, it's also easy to have a proper amount of betaine intake as food, because there are food that are particularly rich of this molecule. There are beets, peanuts, seafood, and so on. And why this molecule is so important? Because it has two classical physiological role. That one is to be important for osmoregulation, so it's considered what is called the compatible osmolyte. That means a molecule that was doing nothing otherwise that counting as osmolyte entity, but this is not true, as I will show you in a few slides. And also for his role of methyl donor, that is another physiological role, and also the role for which was approved as drugs, because is involved in homocystinuria, and for that it can be, we can say that there are a safe intake till 15 grams a day, but usually with food we are around, as point here, from 1 to 2.5 grams a day, simply with a normal diet. In looking to literature, betaine moves across the cell membrane through these four transported, but in reality, as you will see, also GAT1 is involved. Another important point to take in mind is that the concentration of betaine is quite high. So it can reach 100 millimolar in kidney, where probably its function as osmolyte is the most important, and 30 millimolar in liver, and instead it is not clear how much betaine we have in the brain. This is because betaine is something that moves inside, outside the cells, can be produced by the cells itself, and so it's not so easy in a so peculiar tissue to be quantified. So look into literature again, you can find some interesting effects of betaine, in particularly positive effect in neurological disorder, and all of these disorder can not be related simply to the fact that betaine works as a osmolyte or methyl donor, because, for example, you see there are these two papers. This is was published in 2022, and this was 10 years before, if I remember correctly, that suggests that betaine is a activator of a ligand-gated ion channel in C. elegans. But recently, looking to what betaine is doing, we also found that betaine has been demonstrated to increase the tolerance in hypertonic thermal stress. And in this review, they reported that there are some evidence that betaine does not simply work as a compatible osmolyte, but also works as a chaperone, so seems to improve the refolding of protein that can be denaturated by excessive heat. So the question is if there are all these points that underline the beneficial effect of betaine, how can betaine reduce the excitotoxicity? Is betaine probably is betaine interacting with the GABAergic pathway? With this in mind, we start to look about the possible role in imbalance and in excitatory inhibitory imbalance. In this situation, where usually we reach a condition of excitotoxicity, if the alteration is due to an increase in excitatory neurotransmission, it's easy to look to for example, to the glutamate transporter as impaired means of cleaning the synaptic cleft. So in this case, the increase of activity is due to a disappearance of glutamate transporter or reduction of glutamate transporter activity. So one goal could be increase the glutamate reuptake. In this situation, the glutamate is reuptaken mainly by the astrocyte. Instead, when we have an excitotoxicity that can be due to a decrease in neurotransmission, in this case, one possibility is to increase the inhibitory neurotransmitter in the synaptic cleft. But this can happens by blocking the transporter, and blocking the transporter could impair the following synaptic release, because if in many cases GABA and also glycine are reuptaken in the synaptic terminal and reused, so this could not be the right choice to modulate and to increase the inhibitory neurotransmission. So we focus on SLC6A1 mutant, mainly with experiment conducted in Xenopus levi-oocytes that are the main cell models used in our lab. These are a very peculiar model that let you to, for example, express by heterologous expression a very high concentration of the transporter, or transplant membrane coming from human or animal specimen, and usually we pair the electrophysiological recording with chromatography or spectrometry measurement of the amount of the substrate. But also we have developed some other assays that involve fluorescence that are not, in this case, are not related to SLC6A1 study. So in the data that I'm going to present today are mainly collecting the biophysical the biophysical characteristics of the transporter, mainly this pre-steady state current that is related to the first step of the transport cycle. For example, when sodium is able to bind to the transporter inside the membrane electric field. So this binding usually causes a change in the amount of charge that you have in the membrane, and so you can quantify the sodium interaction and the way that this sodium binds. So also this information can also be related to if you study mutants, for example, if there was a change in the kind in the position of the binding on sodium. And then you can also collect the classical transport associated current, because GAT1 is an heterogenic transporter. So from this you can also make some analysis and get QV relationship, IV relationship, and so on. These data are the data that I already presented in July in one of the online meeting, but just to show, we demonstrated that rat GAT1, but also human, is able to transport betaine with a classical dose response and with a classical voltage dependence. The transporter is blocked by SKF, is completely absent in the absence of sodium. So all this point suggests that betaine is clearly transported by GAT1. So and also we collect the data measuring the amount of GABA and betaine that goes inside the cells when we expose the oocytes to the two compounds. Okay, but oops. The most interesting point is the relationship between GABA and betaine. So if we perfuse these two compounds at different concentration, there are at some point the betaine starts to impair the transport of GABA. And this happens in a specific concentration that are the concentration that correspond to the around the K-alpha of GABA for GAT1 and the K-alpha of betaine for GAT1. To better show and to better visualize this point here is, I think, something like 64 different conditions with at least 5 oocytes for each measurement with many different concentration of GABA and betaine. And, okay, oops, sorry. Okay, if you look here, there is this, I don't know if you are able to appreciate with in this slide, but here you have a white zone. So if you see, for example, when you have GABA 3 millimolar, the current increase if you go from 1 to 3 and to 10, but then if you move, increasing the
only thing I ever wanted in life was to be a mother.
the concentration of betaine, there is a point where the current became very, very small. And this is the point around 0.1 and 0.3 millimolar betaine. And this is a very, how to say, specific event. So, and so we tested and to better determine this data, and we confirmed this dual behavior of betaine. It works as a GABA inhibitor with a EC50 around 7 micromolar, and instead it became a slow substrate with a K-alpha of around 9 millimolar. This role was confirmed also using LC-MS uptake, and but we wonder why, how could betaine selective modulate the GABA uptake by GAT1? We think that this happens because betaine from our data seems to be one of the slow substrate, and this slow substrate can be a particular molecule that can interact and change the activity of the transporter. So it's something that can be shown better if we look to these IV curves. Here we have, for example, the nice current that we get with betaine and the current that is obviously larger, but this happens also with the betaine transporter BGT1. But when you decrease your concentration, you go to the value that I showed you before, even at very negative potential, your current is completely blocked by the presence of betaine. And I don't want to spend too much time to explain these slides, but this is the slide that shows how we have studied the pre-steady state. So this is a GATT wild type in presence of sodium. This is when we block completely the transporter. And this is what happened when we add GABA at the saturating concentration. Here, instead, you see that if I add an amount of GABA that is close to the K-half, we can partially see again some of this pre-steady state. And similarly, we see this pre-steady state when betaine is present at high concentration. That probably is not the saturating one, but the transporter is really slow. And moreover, I can go up to the next slide. Here it became more evident, even for the people that is not used to this biophysical characterization, that this is the current in presence of GABA 10 millimolar. These are the transient. And if I add beta into 100 micromolar, the current is greatly reduced, but the transient became, if possible, much larger. We analyze this transient, and what we conclude is that the pre-steady state are really slowed down by the presence of betaine. So betaine slow down the transport cycle and seems to inhibit the GABA binding and translocation. So the transport in this case is not completely disabled, but simply does not work at that kind of concentration of betaine. This means that if we don't have enough GABA around, betaine can act and blocking the transporter and let the amount of GABA increase. As the amount of GABA increase, the effect of block given by betaine stops to be there, because if you go up the 10 micromolar, everything go back to the normal. Instead, if you go up with betaine concentration, betaine became another substrate. Moreover, in the presence of betaine, the K-alpha for sodium, the affinity for sodium changed. So the presence of betaine require much more sodium than GABA alone. Oops, okay. Now, these are, if I reach the slide, okay. These are the new results. These are mutants that we study in the past. So some, for example, are not mutant related to the disease. But we decided to study mutants that were already well known for the K-alpha for GABA and for the K-alpha for sodium in order to understand better the action of betaine. So we select these mutants that has a very, very low GABA K-alpha, so and very high affinity. And in this case, we see a stronger betaine inhibition. You see here is the transport is completely blocked, but then again, in the presence of a higher concentration of betaine, there is a cumulative effect of GABA and betaine. So this is a wild type that I already showed, and then we move to another transporter that has a higher K-alpha compared to the wild type, and here the block is less strong, but is moved through the more higher value, to block the transporter you need higher value of betaine. And here, where you have a very high K-alpha of GABA, seems that betaine is doing nothing. So this is the same representation that I showed you before, and you have to focus. Okay, this is not the, okay, so I removed this, I changed in the last presentation, but it's not a problem, so I can anyway explain. If you look here, this is a mutants with a very low GABA affinity, high GABA affinity, and the white dot move in closer to the lower betaine concentration. This is the wild type. And so you see that this is confirmed. This kind of action of betaine is confirmed even if a different concentration related to the characteristics of the mutant. Another point that is important to underline is that when you have a mutation, and we know from these three examples that we show, the mutation can change the pH dependency. GAT1 wild type is not pH dependent, and is not pH dependent even when tested with betaine. The data with GABA has been already published many years ago. But no, okay, sorry, here, sorry. The story is that GABA wild type is not pH dependent when tested with GABA, but became pH dependent when you test it with betaine. So that means that probably, as we thought, betaine needs much more gradient to go inside the cells, and here probably the current is increased because GAT1 is using proton instead of sodium. And this and the relation, the T349 and the K443 that have a opposite pH dependency demonstrate to do the same also in the presence of betaine, and instead these mutants is not sensitive to the pH. It's important to look to this aspect not only for betaine but in general for the mutated transporter, because the changes in pH dependency can greatly change the transport efficiency. So these are the conclusion for the wild type. So the betaine can protect the neurons from outside, slowing down the transporter, increasing the GABA viability when is needed, so when there are few GABA around, and also from inside, because from what you know and what we want to investigate better, betaine can probably do something also in ameliorate the expression of the transporter. So the conclusion is that betaine at millimolar concentration can enter in the neuron. Betaine is a slow substrate of GAT1. In the presence of betaine at micromolar concentration, the transport of GAT is impaired when GABA is below the K-half. Betaine, independently by the concentration, slow down the first steps of the transport cycle, and so we have seen that it increased the decay time, affect the transport rate, and modify the QV relationship, means that modify the interaction of the transported with sodium. And also, this is supported by the change in the K-half. And when betaine instead, when betaine is tested on the mutants, can act differently. It depends about the changes that you have in mutant characteristics and mutant interaction with GABA. So the K-half for GABA of each mutant can make each mutation in the disease different. And it's important to underline that like the wild type, when the transport of GABA is impaired in the presence of betaine. But in case of the mutants, the value of the relationship between the concentration of GABA of betaine changes. And the functional mutant often change also the K-half for sodium. This, again, can change the action of betaine. And so, as I say, the betaine action can be completely different according to the mutation. So, at this point, we cannot say if betaine can be beneficial or not for any mutation, for SLC6A1 and DD in general, or can be beneficial only for some mutants or cannot. And so what I want to underline, similarly for substrate and any other drugs that acts on the functionality of GAT1, need to be investigated on specifically on that mutation. Okay, so we have still a lot of open questions. The first is because no one had looked for betaine inside the synaptic vesicle, and this is something that we have to organize our lab to do, but if someone wants to collaborate and to help us to find out and to answer to this question, the blue questions are the ones that are open for collaboration. We are testing if betaine can act as chaperone for mutated membrane protein, because the data that reported in literature shows that betaine can act as chaperone, but for cytoplasmic membrane protein. And we are also interested to understand what are the determinants involved in this dual role of betaine. We have looked into the biophysical data. We excluded the fact that could be an allosteric modulation. Is something that interacts in the binding site. And so we wonder if betaine is really trapping one sodium, and we are close to give this answer, and that could be yes. And we know that betaine, if this is true, it means that betaine require high couple energy to be moved inside the transporter. Another question that is open, we are not working with animal models, so we are not able to answer to this question. Can betaine concentration be increased in the brain by betaine supplementation? And these two questions are work in progress in our labs. So we are studying if betaine acts similarly also on the other GABA transporters. It could be quite interesting. And finally, we are now analyzing the action on GABA receptor, and there is a poster of one of my postdocs about that. And also, the second poster coming from my lab is instead about the characterization also with betaine of one of the mutation that causing the SLC6A1 disease. Okay, with this I want simply to thank my collaborator, all my lab, and particularly Manan Bhatt, that was the former PhD student that start the story of betaine, together with Tiziana Romanazzi and Angela Di Jacopo, that are here with are now also the two postdocs and are here with the poster. The precious technician, Raffaella Cinguetti, my colleague Cristina Rosetti, and the two new PhD students, Chiara D'Agostino and Giulia Casoli. And here are the collaborators that are part of the Neurotrans project, and all the other collaborators at the university. And you all, and Amber in particular, for inviting me here. So thank you.